This session describes the autonomic nervous system neurotransmitters and receptors. The learning objectives are to review the anatomical and functional classification of the autonomic nervous system, to describe the autonomic nervous system neurotransmitters and receptors, and to describe the central control of the autonomic nervous system. Remember that the autonomic nervous system is the portion of the nervous system that controls the visceral function of the internal organs of the body. It's organized on the basis of the reflex arc, and it's formed of sympathetic division that prepare the body to face fight and fright and emergency situation, and a parasympathetic division that is dominant during rest and digestion. The sympathetic division of the autonomic nervous system, its nerves originate from the lateral horn cells of all the thoracic and the upper three lumbar segments of the spinal cord. So it's called the thoracolumbar system. The parasympathetic division of the autonomic nervous system is the craniosacral outflow of the autonomic nervous system. The cranial outflow supplies the visceral structure in the head and neck via the oculomotor, the third cranial nerve, the facial, which is the seventh cranial nerve, the glossopharyngeal nerve, which is the ninth cranial nerve. About 75% of all the parasympathetic nerve fibers are in the vagus, which is the 10th cranial nerve, passing to the entire thoracic and abdominal viscera. For the sacral outflow, it supplies the pelvic viscera via the pelvic branches of the second, third, fourth sacrospinal nerve. So remember that the autonomic nervous system is organized on the basis of the reflex arc. It controls the involuntary plane muscles, the heart, and the glands. Each autonomic involuntary pathway is formed of two fibers, preganglionic fibers and postganglionic fibers. So it's a two-neuron system. Now coming to an important question. What are the neurotransmitters of the autonomic nervous system? It's important to know that in autonomic nervous system, transmission at the synaptic junction between the pre- and postganglionic neurons and between the postganglionic neurons and the autonomic effectors are chemically mediated by the neurotransmitters. There are two main neurotransmitters involved, acetylcholine and noradrenaline. Also remember that in the somatic nervous system, acetylcholine is the neurotransmitter. On the basis of the neurotransmitter release, the autonomic fibers are divided into cholinergic fibers and adrenergic fibers. At the same time, the receptors which are activated by acetylcholine are called cholinergic receptors, and those activated by noradrenaline are called adrenergic receptors. Starting with acetylcholine. It's the neurotransmitter released by the cholinergic fibers. Cholinergic fibers are present in all preganglionic autonomic fibers, including all preganglionic sympathetic fibers and all preganglionic parasympathetic fibers. Acetylcholine present in the preganglionic sympathetic fibers to the adrenal medulla, the modified sympathetic ganglion. On stimulation by acetylcholine, it releases adrenaline and noradrenaline. Acetylcholine also released from all postganglionic parasympathetic fibers. Adding to this, acetylcholine is released from some postganglionic sympathetic fibers that are secretory fibers to the sweat gland and vasodilator fibers to the blood vessels of the skeletal muscles. Now, acetylcholine synthesis, storage, and release. Acetylcholine is sensitized in the terminal endings of the cholinergic nerve fibers. The termination of any nerve fiber have several enlargements called synaptic knobs. These knobs are rich in mitochondria to supply the ATP required for the synthesis of the neurotransmitter. Also, they contain the transmitter vesicles which contain the neurotransmitter. Synthesis of acetylcholine involves the reaction of choline with acetate. Choline is synthesized in the neuron, and also there is an active uptake of choline into the cholinergic neurons. Acetate is first activated by the combination of acetate group with the reduced coenzyme A to give the active acetate, the acetyl-CoA. Acetyl-CoA and choline in presence of the choline acetyl transferase will give us acetylcholine. Most of the synthesis occur in the exoplasm, and most of the acetylcholine is then transported to be stored in clear vesicles in the termination of the cholinergic fibers. When an action potential spreads over the terminal fibers, the depolarization process increases the permeability of the fiber membrane to calcium ions, thus allowing these to diffuse in moderate number into the nerve terminals. They interact with the vesicle adjacent to the membrane, causing them to fuse with the membrane and to empty their content to the exterior. Thus, acetylcholine is released by exocytosis. Once acetylcholine is released, it binds with the cholinergic receptor and produces the effect of acetylcholine on the effector organs. Once acetylcholine has been secreted by the cholinergic nerve endings, most of it within a fraction of a second splits into acetate ions and choline by the enzyme acetylcholine esterase. We have two types of acetylcholine esterase: the true or the specific choline esterase that has a high affinity for acetylcholine and is present in high concentration in the membrane of the cholinergic nerve endings. The pseudocholine esterase, which is non-specific and is present in the plasma and acts on the acetylcholine that diffuses into the surrounding fluids or into the plasma. The choline that is produced is transported back to the terminal nerve endings, where it is used again for synthesis of a new acetylcholine. Once acetylcholine is released, it binds with a specific receptor, the acetylcholine receptor, known as cholinergic receptors. Acetylcholine activates two different receptor types, muscarinic and nicotinic receptors. The reason for these names is that muscarine activates the muscarinic receptors only and will not activate the nicotinic receptor, whereas nicotine in small dose will activate only the nicotinic receptors. Nicotinic receptors are classified into nicotinic receptor number 1, which is present at the motor end plate in the neuromuscular junction, and nicotinic receptor number 2, which is present in the autonomic ganglia and the adrenal medulla. The muscarinic receptor, on the other hand, are classified into 5 different types of muscarinic receptor present in the effector organs. Remember that the nicotinic receptors are stimulated by small dose of nicotine and inhibited by large dose of nicotine. The muscarinic receptors are stimulated by muscarine and inhibited by atropine. Now, the mechanism of action of the cholinergic receptors. Once acetylcholine binds with its cholinergic receptor, at the level of the nicotinic receptor, there will be increase in the intracellular sodium and calcium, while at the level of the muscarinic receptor, the mechanism varies according to the type of muscarinic receptor. It may be increase in the inositol triphosphate or diacylglycerol or the intracellular calcium, or there may be inhibition of the adenyl cyclase, thus decreasing the cyclic AMP. Once acetylcholine binds with its cholinergic receptors, the action of the acetylcholine will be manifested as the parasympathetic activity present in different parts of the body, which is dominant during rest and digestion. Now, the second neurotransmitter of the autonomic nervous system, which is the norepinephrine. It's the neurotransmitter released by the adrenergic fiber, which include all the post-ganglionic sympathetic fibers except the secretory fibers to the sweat gland and the vasodilator cholinergic fiber to the blood vessels of the skeletal muscle. Remember that the adrenal medulla has the ability to secrete epinephrine and norepinephrine. Now, norepinephrine synthesis, storage, and release. Norepinephrine is sensitized in the terminal endings of the adrenergic nerve fibers. It's also secreted from the adrenal medulla with adrenaline. In the exoplasm of the adrenergic nerve fibers, the amino acid tyrosine by hydroxylase enzymes change into DOPA. DOPA by decarboxylase enzyme change into dopamine. dopamine by the hydroxylase enzyme change into norepinephrine. Norepinephrine is stored in granulated dark vesicles bound to the ATP and a protein called chromogranin. When the nerve impulse reaches the termination of the adrenergic nerve fibers, calcium influx occurs, leading to exocytosis of norepinephrine that bind on the postsynaptic membrane with its specific adrenergic receptors that include beta-1, beta-2, and alpha-1. Also, it binds with alpha-2 in the presynaptic membrane. Binding of norepinephrine with its adrenergic receptor will produce its effect on the effector organ. Following the secretion of norepinephrine by the terminal nerve endings, it's removed from the secretory site within few seconds in three different ways. Either reuptake into the adrenergic nerve endings themselves by the active transport, and this accounts for the removal of 50 to 80% of the secreted norepinephrine, or the diffusion away from the nerve endings into the surrounding body fluid and then into the blood, or destruction by the enzyme, either oxidation by the monoamine oxidase or the methylation by catechol or methyl transferase. Note that in adrenal medulla, the N-methyl transferase change norepinephrine to epinephrine. That's why the adrenal medulla has the ability to secrete both norepinephrine and epinephrine. Both together are known as catecholamine. It's also important to know that the N-methyl transferase enzyme is not found in postganglionic sympathetic nerve fiber. It's only found in the adrenal medulla and some areas of the CNS. That's why the postganglionic sympathetic nerve fiber secretes only norepinephrine. Always remember that norepinephrine is a neurotransmitter and a hormone secreted by the postganglionic sympathetic nerve fiber and the adrenal medulla, while the epinephrine is the major hormone of the adrenal medulla, cannot be secreted by the postganglionic sympathetic nerve fiber because it doesn't have the N-methyl transferase enzyme. Norepinephrine binds with its specific receptors known as adrenergic receptors. Research experiments using different drugs on effector organs supplied by adrenergic fibers shown that there are two major types of adrenergic receptors, alpha and beta receptors. Two subtypes of alpha receptors, alpha-1 and alpha-2, and three subtypes of beta receptor, beta-1, beta-2, and beta-3. The adrenergic receptors are present on the surface of the effector organ, and they are called postsynaptic receptors. Also present in the membrane of the postsynaptic nerve endings, and they are called presynaptic receptors. The mechanism of action of the adrenergic receptors. Once norepinephrine binds with alpha-1 receptor, produces their effect through increasing the intracellular calcium, while binding of norepinephrine with alpha-2 receptor produces their effect through inhibiting adenyl cyclase and thus decreasing the cyclic AMP. On the other hand, when norepinephrine binds with the beta-adrenergic receptors, beta-1, beta-2, or beta-3, produces their effect by stimulating the adenyl cyclase and thus increasing the cyclic AMP. The adrenergic receptors are distributed on different organs and have different functions. Stimulation of alpha-1 receptor present in the postsynaptic membrane of the blood vessel, the GIT sphincter, the internal anal sphincter, and the internal urethral sphincter will lead to vasoconstriction, contraction of the GIT sphincter, contraction of the internal anal sphincter, and contraction of the internal urethral sphincter. Stimulation of alpha-2 receptor present in the presynaptic membrane will lead to inhibition of norepinephrine release, thus providing a negative feedback. Stimulation of beta-1 receptor present in the cardiac muscle will lead to increase in all cardiac properties, while stimulation of beta-2 receptor present in the postsynaptic membrane of the ciliary muscle, of the bronchi, of the GIT wall, of the rectal wall, and the urinary bladder wall will lead to relaxation of the ciliary muscle, thus provide a far vision, relaxation of the bronchi, relaxation of the GIT wall, relaxation of the rectal wall, and relaxation of the wall of the urinary bladder. Stimulation of beta-3 receptor present in the adipose tissue will lead to lipolysis. Once the catecholamine, epinephrine, and norepinephrine bind with their adrenergic receptors, the action of the catecholamine is seen in the body as sympathetic activity of different parts of the body, preparing the body to face fight, fright, and emergency. Now, let's apply what we discussed about the adrenergic receptor to see the effect of sympathetic nervous system on different parts of the body. The effect of sympathetic nervous system on the eye. Stimulation of beta-2 adrenergic receptor present in the ciliary muscle leads to relaxation of the ciliary muscles of the eye, accompanied by stretch of the suspensory ligament and flattening of the lens, thus preparing the eye for the far vision. At the same time, stimulation of alpha-1 adrenergic receptor present in the radial muscle of the iris leads to contraction of the dilator pupillae muscle, thus producing mydriasis. The effect of sympathetic nervous system on the heart. By stimulation of beta-1 adrenergic receptor, there is increase in all cardiac properties, excitability, rhythmicity, conductivity, and contractility, manifested on the patient as increase in the heart rate and the arterial blood pressure. The effect of sympathetic nervous system on the lung. Through stimulation of beta-2 adrenergic receptor present in the wall of the bronchi lead to relaxation of the smooth muscle present in the bronchi, thus producing bronchodilation. At the same time, stimulation of alpha-1 adrenergic receptor present in the wall of the blood vessel produces vasoconstriction. That's why sympathomimetic drugs are used in treatment of bronchial asthma. Always remember that stimulation of beta-2 adrenergic receptors produce relaxation of the smooth muscle. Example of this, stimulation of beta-2 adrenergic receptors present in the wall of the bronchi produce bronchodilation, while stimulation of alpha-1 adrenergic receptors produce contraction of the smooth muscle. Example of this, stimulation of alpha-1 adrenergic receptors present in the wall of the blood vessel produces vasoconstriction. The effect of sympathetic nervous system on the GIT. By stimulation of beta-2 adrenergic receptor present in the wall of the GIT produces relaxation of the wall of the GIT, while stimulation of alpha-1 adrenergic receptor present in the GIT sphincters leads to contraction of the sphincters. The effect of sympathetic nervous system on the rectum and the anal sphincter. Stimulation of beta-2 adrenergic receptor present in the rectal wall produces relaxation of the wall of the rectum, while stimulation of alpha-1 adrenergic receptor present in the internal anal sphincter leads to contraction of the internal anal sphincter. These two effects will lead to retention of the stool and inhibition of defecation. The effect of sympathetic nervous system on the urinary bladder and the urethral sphincter. Through stimulation of beta-2 adrenergic receptor present in the wall of the urinary bladder leads to relaxation of the wall of the urinary bladder, while stimulation of alpha-1 adrenergic receptor present in the internal urethral sphincter leads to contraction of the internal urethral sphincter. Both these effects will lead to retention of the urine, thus inhibition of micturition. Now, coming to an important question. What is the control of the visceral reflexes of the autonomic nervous system? That's to say, what is the central control of the autonomic nervous system? The answer to this is that the levels of the autonomic integration within the central nervous system are variable. Simple reflexes are integrated in the spinal cord, such as urination, defecation, erection, and ejaculation reflexes. More complex reflexes are integrated at higher level in the central nervous system. Complex reflexes that regulate respiration and blood pressure are integrated in the medulla, while reflexes that control the pupillary response to the light and accommodation are integrated in the midbrain, thus to say the reticular formation of the brainstem. Complex autonomic mechanisms that maintain the chemical composition and the temperature of the internal environment are integrated in the hypothalamus, while the emotion and behavioral reflexes are regulated by hypothalamus, limbic system, and the cerebral cortex. By this, we finish today's session. I hope you enjoyed it. Thank you.